Hi, Emma. What are the primary differences between plant and animal cells?
Great question. Both plant and animal cells are types of eukaryotic cells, meaning they have a nucleus and other membrane-bound organelles. However, they do have key differences. Let's start with size. Plant cells typically range from 10 to 100 micrometers, while animal cells are usually between 5 and 50 micrometers.
So, what makes up the structure of a plant cell?
In plant cells, the structure is strongly defined by the cell wall, which is made up of cellulose and sometimes other components like hemicellulose and pectins. The cell wall gives the cell its shape and supports the entire plant structure. Additionally, plants have something called plasmodesmata, which are small channels that allow for communication and transport of substances between cells.
I've also heard about chloroplasts in plant cells. Can you explain those?
Absolutely. Chloroplasts are unique to plant cells and are where photosynthesis occurs. They contain their own DNA and are thought to have originated from a symbiotic relationship with cyanobacteria. This is a great example of the endosymbiotic theory, where an early eukaryotic cell took in a photosynthetic organism, leading to the evolution of chloroplasts.
And what about mitochondria? Do both plant and animal cells have them?
Yes, mitochondria are found in both plant and animal cells. They're the powerhouses of the cell, generating energy through cellular respiration. They also have their own DNA, supporting the theory that they, too, originated from an ancient symbiotic relationship.
What are the unique features of animal cells, then?
Animal cells have structures called lysosomes, which are enzyme-filled organelles responsible for breaking down waste materials within the cell. Additionally, animal cells have a flexible plasma membrane that contains cholesterol, giving it fluidity and allowing the cell to change shape as needed. Unlike plant cells, animal cells don't have a rigid cell wall, which gives them a more flexible, often rounded shape.
I see. And how do plant and animal cells divide?
Good observation. Plant cells divide by forming a cell plate, also known as a phragmoplast, which grows outward until it splits the cell into two. Animal cells, on the other hand, divide through a process called cytogenesis, where the cell membrane pinches inward, eventually splitting the cell in two.
So, what are some of the foundational differences between plant and animal cells?
That's a good starting point. Both plant and animal cells are eukaryotic, meaning they have complex structures, including a nucleus. However, there are significant differences. Let's start with plant cells. They're typically larger, around 10 to 100 micrometers, while animal cells range between 5 and 50 micrometers.
Interesting. What makes plant cells so unique?
Plant cells have a rigid cell wall made primarily of cellulose, but it can also include other materials, such as hemicellulose and pectins. This wall gives structural support and helps the plant maintain its shape. Plants also have something called middle lamella, a pectin-rich layer that acts as a glue between adjacent plant cells.
I've read about different types of plant tissues too. How are these structured?
Exactly. Plant tissues are made up of various cell types that each play specific roles. Parenchyma, for instance, is a type of plant tissue that's involved in functions like photosynthesis, storage, and regeneration. Then there's collenchyma, which provides structural support, especially in areas of the plant that are still growing. Another type, sclerenchyma, is very rigid and helps support mature parts of the plant, and it's strengthened by a secondary cell wall.
And what about those chloroplasts? I know they're important for photosynthesis.
Yes, chloroplasts are fascinating organelles. They are semi-autonomous, meaning they contain their own DNA and can replicate independently. Chloroplasts are where photosynthesis occurs, converting light energy into chemical energy stored in glucose. Interestingly, these chloroplasts are thought to have originated from an ancient symbiosis with cyanobacteria.
So, they were once independent organisms?
That's the idea. It's based on the endosymbiotic theory. This theory suggests that certain organelles, like chloroplasts and mitochondria, originated from free-living bacteria that were absorbed by early eukaryotic cells. Over time, they became integral parts of the cell.
Speaking of mitochondria, both plant and animal cells have them, right?
Correct. Mitochondria are known as the powerhouses of the cell because they generate energy through cellular respiration. They are also semi-autonomous with their own DNA, just like chloroplasts. Plant cells use mitochondria to produce energy, especially in areas where photosynthesis can't meet the cell's energy demands.
Do animal cells have any unique structures?
They certainly do. Animal cells lack a cell wall, which gives them flexibility. They do have lysosomes, which contain enzymes to break down waste materials through a process called hydrolysis. Animal cells also contain centrioles within a structure called the centrosome, which is essential for cell division.
So, centrioles are crucial for cell division?
In animal cells, yes. Centrioles organize microtubules during cell division, helping to separate chromosomes accurately. However, most plant cells don't have centrioles. Instead, they rely on other microtubule organizing centers, showing that centrioles aren't strictly necessary for cell growth.
I've heard that plant and animal cells respond differently to their environments. Can you explain that?
Great point. In a hypertonic environment, where there's a high concentration of solutes outside the cell, animal cells shrink in a process called plasmaryza, whereas plant cells retain their rigid shape because of the cell wall. However, the living part inside the plant cell, called the protoplast, will shrink away from the wall, a process called plasmolysis. In a hypotonic environment, where water concentration is higher outside the cell, an animal cell may burst due to osmotic pressure. Plant cells, however, can take in water and swell, but the cell wall prevents them from bursting. This pressure buildup, known as turgor, helps maintain plant structure.
That's a lot of information. What about the extracellular matrix? How does that fit in?
The extracellular matrix, or ECM, is essentially the noncellular component present within tissues and organs. It's particularly important in animal cells, where it provides structural support and facilitates cell communication. ECM is rich in proteins like collagen, which provides tensile strength, and proteoglycans, which retain water and form a gel-like structure that supports cellular adhesion.
So, ECM is mostly for structural purposes?
Not solely. ECM has diverse functions. For example, it plays a role in cell migration, wound healing, and even metastasis of cancer cells. Certain proteins in the ECM, called integrins, anchor cells to their surroundings and enable communication. When these integrins bind to specific ligands in the ECM, it triggers a signal cascade within the cell, influencing processes like adhesion, migration, and endocytosis.
Are there any conditions linked to the ECM?
Absolutely. ECM-related pathologies often arise from mutations in genes that code for its components. For instance, defects in collagen formation can lead to connective tissue disorders. Osteogenesis imperfecta, or brittle bone disease, is one such condition caused by mutations in collagen type I. Another example is Ehlers-Danlos syndrome, where faulty collagen results in overly elastic skin and hypermobile joints.
And what's the role of elastin in the ECM?
Elastin is another protein found in the ECM, especially in tissues that need to stretch and then return to their original shape, like skin, blood vessels, and lungs. Elastin molecules allow tissues to maintain elasticity, which is vital for organs that frequently expand and contract.
Fascinating. Does ECM have any role in the immune response?
Yes, indirectly. For instance, certain ECM components, like fibronectin, act as scaffolds for cells during wound healing. Fibroblasts are cells that produce ECM, and in cases of tissue injury, they are particularly active, producing more ECM to support the healing process.
What happens if there's a mutation in integrins or other ECM-related proteins?
Mutations in integrins or other ECM components can lead to inadequate cell adhesion, which might cause cells to detach too easily. This is a mechanism behind certain cancer cells that lose their attachment to primary tumors and travel to other parts of the body. Additionally, defective ECM interactions can impair wound healing or immune cell function.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.